Hello, madam.
Hello.
I'm audible, madam?
Yeah.
Yeah. So you're audible, madam. So can you share your presentations for testing?
Sure.
Yeah. Yes, it is good, madam.
Okay.
It's visible to us.
Okay.
You can stop now, sir. Yeah, stop, madam. Yeah. Thank you, madam.
Good evening, everyone, and welcome to today's webinar on immunity in today's world, hosted by the Department of Biochemistry, St. Philomena's College, Mysore. I, Dorin D'Souza, Assistant Professor of BWalk Healthcare Technology, will be your host for today's webinar. We are on the final day of the webinar, and before we go further, I would like to review through the functionality to go through the webinar. Your active participation is very important throughout the session, so I request you all to maintain the stable internet connection throughout the session. If you have any questions, kindly post them in the chat box. You can enter your questions as well as your comments in the chat box throughout the presentation. All your questions will be answered by the speaker at the end of the session. We will send all the relevant links after the event, which includes the feedback form, and it is mandatory to fill in the feedback forms in order to generate your e-certificates. Now we shall begin our first session for the evening. Our speaker for today is Dr. Akila Prashant, Professor, Department of Biochemistry, JSS Medical College, Mysore, who is going to present on the topic COVID-19 and vaccines. I request Ms. Shruti, Assistant Professor, Department of Studies in Biochemistry, St. Philomena's College, to introduce the speaker to us.
Thank you, ma'am. It's a privilege for me to introduce our distinguished speaker, Professor Akila Prashant from the Department of Biochemistry, JSS Medical College, Mysuru. Madam completed her MBBS in 2000 from the University of Mysore and did her MD in Biochemistry from Manipal University. Then got her postgraduate diploma in Genomic Medicine and Healthcare in 2018 from University of South Wales. She owns a PhD degree from JSS Academy of Higher Education and Research. Molecular biology and genetics are her areas of research interest. In recognition of her research work, Madam has been awarded with the prestigious Professor Y.T. Tatachari Endowment Prize for the Best Research Scholar at the Graduate Ceremony 2015 at JSS Medical College, Mysore. She has given a major contribution in the development of the Department of Medical Genetics, JSS Medical College. She has authored 40 publications in reputed journals like Clinica Chemica Acta, PLOS One, Indian Journal of Clinical Biochemistry, J. Dermatol Science, Reports in Biochemistry and Molecular Biology. With this brief introduction, we welcome you, ma'am, and I request you to take over the session.
Thank you, Shruti, for this kind introduction. I'll be sharing the screen right now. Yeah, so at the outset, I would like to thank the organizers for giving me this opportunity to share some of the knowledge which we have gathered through with a lot of review of literatures regarding the COVID-19 and its vaccine candidates. It's quite early to actually, talk about this because the disease as such is a new one, and talking about vaccines is something in a very infant stage. I should say it is in a very infant stage. Okay. With this note, probably the content of the today's talk would be, I will start with a brief introduction of the body's defense mechanism. I'm sure everybody would have gone through this in the past 2 days. You would have had a brainstorming session on this. I'll be touching upon this in a very brief manner because vaccines actually work on this defense mechanism. So it is very important for us to know this before going to the vaccine as such. Okay, then we come up with the road to the vaccine development, how the progress happens in the area of the vaccine development. Then we will see how the COVID-19 vaccine has started with the it has reached up till now, till whatever pace it has come up till now is quite in a pandemic speed as such. And then we'll discuss different vaccine platforms which are available and some of them which have reached the phase 3 trials, some candidate vaccines which have currently reached the phase 3 trials, and the challenges and the future directions in this area of vaccine development. And with this, probably I would, at the end of this talk, probably I could inspire some of the students and make an impact on them so that the young minds can think much beyond their regular curriculum. So coming to the basics, how the body responds to the foreign antigens or any viral insult which happens to the body, how does our immune system reacts to it? So we have a process of humoral immunity, and there is something which is called as cellular immunity. So in humoral immunity, what happens is when the B cells encounter an antigen, the antigen is presented to the B cells, and the B cell receptor recognizes this antigens, and it engulfs the antigen by the process of endocytosis. And once the engulfing happens, then in the intracellular place, this lysosome fuses with the phagocytic vesicle and the forming of phagolysosomes. This phagolysosomes then digest this antigen and breaks them into smaller fragments. And the fragments are then displayed on the MS, that is the major histocompatible complex. And then it is presented to the T cells. There is T cell activation which takes place. That is a separate process. Meanwhile, this B cells, they become, they are transformed to plasma cells, and they multiply, they form clones of plasma cells. And these plasma cells are the ones which produce the antibodies which are specific to the antigens. Okay, so this is a process of humoral immunity. Coming to the process of cellular immunity, here, once the T cells are presented with the complex of antigen and MHCs, so the T cells activation takes place. When the T cells are activated, it starts releasing the cytokines. Okay, so the cytokines are released, which will produce the mature T cells, and these T cells will undergo differentiation to form different types of T cells. The different types of T cells, like cytotoxic T cells, these destroy the infected cells. Then we have the helper T cells. These also, they direct some of the other lymphocytes to the site of infection. They help the other cells to arrive at the site of infection. Then we have the suppressor T cells. These inhibit the T and the B cells. Okay, and we have the memory T cells also, which recognizes the antigen for future attack. So this is a basic process what happens whenever an antigen invades our system. This is how the defense mechanism acts to overcome this infection. Now, suppose if our defense mechanism does not overcome the infection, then we are encountered with that infection and we suffer from that disease. So what happens when a vaccine is given? So vaccine, it acts through these defense systems and it helps us to fight against this antigen which has invaded our system. So vaccines, by stimulating the immune system with an infectious agent or the components of the infectious agents, which is modified in such a manner that no harm or disease is caused, but ensures that when the host is confronted with the infectious agent, the immune system can adequately neutralize it before it causes any ill effects. So this is what our vaccine does. So we can see here that when the vaccine is injected into the system, it can be, the route of injection can be anything like intramuscular, intradermal, or subcutaneous, anything. So these vaccines, that is the viral infectious agent or a component of the infectious agent, it stimulates the immune system. Okay, so there will be the activation and the migration of these macrophages and the dendritic cells to the lymph node. In the lymph node, the presentation of this antigens happens, and then the activation of B cells and T cells takes place. The T cells form the cytotoxic T cells. The B cells will transform into plasma cells, producing the antibodies. And of course, some of the T and B cells become memory cells. Okay, so when a person is vaccinated and he confronts with this diseased agent the second time, there will be the defense mechanism which is able to fight against this infectious agent. So we are boosting our immune system. Okay. So where did it all start from? The vaccines. It was Edward Jenner. He's called as a father of immunology. He's a English physician and a scientist who pioneered the concept of vaccines. He created the world's first vaccine against the smallpox. And from then, the vaccines have become the most successful means of preventing any infectious diseases and saving lives every year, because every year we encounter one or the other epidemic. So this has become an important tool by which we can fight these infectious agents. Okay, so here we can see a picture of Dr. Jenner, who is performing his first vaccination on an 8-year-old boy. This is in the year 1796. So this is how the vaccine works. Actually, the same thing which I had told earlier also, and when the vaccine component of the vaccine is injected into the person, it can be either a part of the pathogen or the whole pathogen which is inactivated. It can be administered to the patient, often by injection, and the body produces antibodies to fight these diseases. And if the individual encounters a disease in future, then the antibodies can destroy the infectious agent. Okay, so vaccines reduce the risk of contracting infectious diseases by working with the body's natural defenses, and it builds the protection. Okay, so is the road to vaccine development that simple? I should say no. It is a long, complex, and a costly process. Okay, why? Because it involves many stages in the vaccine development, and it can be classified. These stages can be divided into the basic, the translational, and the clinical development. So what happens in each of these stages? So in the basic stage, it is mainly the discovery and the exploratory stage. Here we study the pathogenic agent in detail. Okay, so here the pathogenesis, how this infectious agent causes the pathogenesis, which immune response it will trigger, and what are the antigens on this pathogenic agent to which the body will show some of the immune responses, and what animal models can be used for studying these diseases in detail. So these are the things which happen in the discovery phase. So some of the candidate which pass this discovery stage, they enter into what is called as the preclinical stage. Okay, so here in the preclinical stage, further testing happens. Okay, the vaccines are designed in such a way, the formulation is formed, and they also test whether the vaccine in its purified form, does it have the same kind of efficacy what it had earlier, or does the efficacy change? Did the stability of the vaccine change? Okay, what happens when the adjuvant is added? did it function the same way as it did before? Okay, so all the proof of concept studies are done. It is studied in the animal models. Okay, then once the candidate passes this preclinical stage, then it enters to the clinical development stage, where we have again 3 phases: phase 1, phase 2, and phase 3. The phase 1, it is done on a small group of individuals, maybe about 10 individuals, where the safety is tested, the vaccine safety is tested, and the dosage of the vaccine. So dose variations are tested in this stage. Then once it passes this stage, then it enters the phase 2 trials, where about hundreds of individuals are tested. And here again, the safety and efficacy of the vaccines are tested in this phase. The candidate vaccines which pass this stage, they enter into the third stage, the phase 3 trials, what is called as. So in this stage, about thousands of individuals are involved in this stage. And again, the same thing, the safety, the efficacy of the vaccines, and then the regulatory approvals also start during this phase. Okay, then once the candidate vaccine passes through all these stages, then the regulatory approval has to be taken. All the documentation has to be submitted to the regulatory authorities. And they, after the review of these documents, and they feel that this vaccine is definitely of need and it is safe to be administered to the human beings, then it comes out to the production phase. And that is the phase 4, wherein the vaccines are, they reach the market, and then the post licensure surveillance takes place. Okay, so this is a very long and complex and costly process. There are several push and pull forces which act on this process. Okay, so typically the timeline which is taken for the development of the vaccine is somewhere between 7 to 20 years. Okay, so it does not happen overnight. It is a very long process, I told you, and definitely a lot of cost involvement is there. Here, suppose if you take it stage wise, because we have seen the stages of different stages in the vaccine development. So stage wise, when we go about for the discovery and the preclinical stage, you know, about 3 to 8 years are gone in this phase, because thoroughly studying of the vaccine, the pathogenicity, the antigenicity, and the targets of where the vaccines can be developed, studying it in the animal models. So all these things takes about 3 to 8 years of time. And then when successful candidates are reported in this stage, then the clinical assay optimization has to be done. That is, when the vaccine is given to a person, the antibodies are developed, those you have to have an assay which can estimate these antibodies. So this assay has to be optimized, and then it comes to the clinical development phase, where it takes about 2 to 10 years of time in the clinical development phase, where it crosses the phase 1, phase 2, and phase 3. Regulatory phase also takes time, about 1 to 2 years. Okay, so the entire process is very lengthy, about 20 years time for the development of one successful candidate. And each phase, there will be like almost 50% reduction in the number of candidates which enters the next phase. So we start with a bigger number of candidate vaccines, but when we end up with the approval stage, we hardly end up with 1 or 2 candidates. Okay, so it is quite an expensive affair. Few examples, if we have to take over the past 50 years, we some of the success stories and some of the failures. Of course, the chickenpox vaccine is a success story, but you can see the number of years it has taken to come up with a successful vaccine. And hepatitis B, of course, so the duration is shorter, and yes, it is a success story. And we all healthcare workers are immunized with the hepatitis B vaccine. So whoever works in the hospital setup, the vaccine is given to them. And recently we got ourselves tested for the antibodies against hepatitis B. And the vaccine has definitely shown a good impact, because we all had the sufficient number of antibodies in our serum when it was tested. So some of these trials are still going on, and some of them have been cancelled because of no successful candidatures which have been across this phase. Okay, so when it comes to development of vaccine towards the COVID-19, definitely it has taken a pandemic speed. We can see when we compare with the traditional the phase of the vaccine development, where each of the phase is, separate. Okay, and it is not, there is no overlapping of the phases, and there is a distinct boundary between each of these phases. But when we come to COVID-19 vaccine development, the COVID-19 vaccine development, all the phases have been merged as such, because there was a need for the vaccine at the earliest. So you can see here the go or non-go decision for the candidate vaccine has been taken up very early. And before the preclinical phase could be completed, the clinical development phase had already started with some of the candidate vaccines. And the human recruitment for the phase 1 trial had begun very early. And some of them have already reached the phase 3 trial in a matter of, maybe 5 months time now. Okay, so once this regulatory pathway is completed, then the large scale manufacturing of the vaccine takes place. Okay, so what was the need for the pandemic speed in the development of coronavirus vaccine? So I think this slide is self-explanatory. You can see how the disease has spread across the globe, and there was a need. This map itself is a month old, I guess, but you can see that how it has affected the entire globe, and everybody were in the wait for, they were awaiting the release of the vaccine. So it had to happen in a pandemic speed. So the need, you can see that the total number of cases confirmed, it is quite a huge number, and the confirmed deaths, it is reached more than 9 lakhs. So this, there was a need for the vaccine to be developed, because this is a public health emergency of international concern. There was a need to break the chain of transmission and limit the spread towards unaffected regions. Of course, now we have very limited unaffected regions, but we do need a vaccine so that we can go about with our normal living. So no specific therapeutic model has been approved to treat and prevent the transmitting infection. So we don't have any specific therapeutic model. So that is a reason, that is one more reason why we require a vaccine. And silent spreaders, they harbor the SARS-CoV virus, and they're equally infectious. So this is also one of the reasons why vaccines are definitely needed. And to protect the high-risk individuals, such as the frontline workers, children, geriatric patients above 60 years of age, and having the comorbid conditions. So we require the vaccines for these individuals. And of course, the entire world is experiencing a serious downfall of economy and global recession. And if we want to go about with our normal lifestyle, probably a vaccine is a much needed thing at this time of the year. So to have the vaccine in place, we have to know the structure of the SARS-CoV. So how this, I'm sure all of you know the structure of this. So we'll just briefly go about the structure. It's a ultra microscopic virus, which has spikes over the outer surface of the envelope. Okay, and this envelope, it consists of a lipid bilayer, which is anchored by structural proteins. We have the membrane proteins. Okay, we have the spike glycoproteins, and we have the envelope proteins. This is the ratio of the membrane envelope to spike, is a ratio of 1:20:300. And E and M proteins are involved in maintaining the structural framework of the virus, whereas the S protein, it is composed of S1 and S2 segments or subunits. So S1 subunit forms the head of the spike, and it is the one which contributes to the immunogenicity, whereas the S2 segment or the S2 subunit, it anchors the spike to the viral envelope. Okay, and genetically, the SARS-CoV-2, it possesses the non-segmental positive sense stranded RNA. It is, the size is ranging between 26 to 36 kilobase pairs in length. Okay, so this is a structure of the SARS-CoV-2 virus. So the latest update from WHO says that about 180 vaccine candidates have been registered with WHO. The latest update what I got in the net was about 180 candidates have been registered in the WHO. Out of this, 35 have been undergoing different stages of human phase clinical trials, and the 145 candidates are in the pipeline of the preclinical evaluation process. Okay, So there are a number of candidates which have been registered with WHO. And this pandemic speed of the vaccine development could happen because of some various tools which we have with us right now. For example, the computational study designs, where without the experimental data, the sequence could be the SARS-CoV sequence was taken and its structure was predicted. Okay, so due to the limited experimental data, several computational study designs based on the existing information were utilized, and to understand the possible molecular mechanisms of the immunological reactions. So once the SARS-CoV genome sequence was released, then they could predict the structural and the non-structural proteins, and which of these proteins can be a target for vaccine development, which was also predicted. And they could predict the secondary and tertiary structures of this virus, the proteins of the virus, the domains, and where they have the substrate recognition sites. So all these things could be predicted. Though I'm not an expert in computational biology, but these are the things which helped in the development of the vaccines. There's also something called as immunoinformatics, which is cost-effective and convenient in silico predictions. This can reduce the number of experiments which are required. So this is all done in silico. For example, the protein sequences from the genome sequences, the protein sequence of the structural and the non-structural proteins were predicted. From this sequence, the antigenicity prediction happened, and there was a development of models for this. Then the epitope prediction from the cytotoxic T cells, the helper T cells, and the B cells, the epitopes were predicted. And there was a in silico vaccine construct which was made, taking in or fusing all these epitopes, that is C cell epitope, the cytotoxic T cell, the helper T cells, and the B cells. All these were epitopes were taken, fused with having the linkers in between. And then they also predicted how this vaccine construct, once introduced into the body, what are the immunological reactions it can trigger in the body, what allergic reactions it can create, what are the antigenic reactions it can induce in the body, how is its stability, what is its sequence homology. All this was predicted. Its secondary and tertiary structures were predicted. And then the molecular docking studies were done, wherein they saw how this vaccine construct, it goes and binds to the toll-like receptors, or the MHC complex 1 and 2. So these are the complexes which will trigger our immune system. So they, by this docking studies, they predicted how it goes and where it goes and binds to these receptors. Then in silico cloning was done, wherein the vaccine construct was uptaken by the vectors, and then its immunological reactions were predicted. So this also helped in coming to a conclusion of which vaccine target can be chosen for the further development. And the details of the immunological pathway of this SARS-CoV-2 was studied in detail, how the virus enters the body, how it binds to the ACE2 receptors, and the entry of the virus happens inside the body. In the respiratory or the airway cells, how the virus replication takes place, the genome replication and the translation of this genome into the proteins, viral proteins, and how it is further released from the infected cells. And these, how that goes and binds the complement activation system, and how it triggers our immune complexes in the body. And it activates the innate immunity system in the defense mechanisms in the body. Suppose if there's maladaptive inflammatory response, then it leads to the cytokine release syndrome. And this accumulation of the neutrophils and monocytes and macrophages happens in the lung tissue, which finally leads to ARDS. So this immunological pathway of the virus was studied in detail, which receptor is involved in the entry of the virus, and which of these antigens bind to this receptors. So these studies were done in detail. The reverse genetic approach also has been used, which have provided the investigators with novel and powerful approach to answer some of the important questions like the biology of the virus, including the function of the viral proteins, their interaction with the cellular host factors, and the mechanism of viral transmission and pathogenesis. So it has also allowed the generation of recombinant viruses, and it's a powerful technology to develop both inactive and the live attenuated vaccines. So this also, all these tools and approaches have helped in this pandemic speed of the vaccine development. So we have different platforms for the development of the different vaccine platforms which have been used in the development of the COVID-19 vaccines. Some of the classical platforms are the whole inactivated virus, live attenuated virus, protein subunits, and virus-like particles. The next generation platforms like the viral vectors, the DNA vaccines, the RNA vaccines, and antigen presenting cells. So both these platforms, that is classical and next generation platforms, have been used in the development of the vaccines. So classical platforms, either the virus-based or the protein-based platforms are used. In the virus-based platform, we have the whole inactivated virus and live attenuated virus vaccines. This whole inactivated virus, they no longer are infectious, they do not replicate, and adjuvants are required to stimulate the immune system. So addition of adjuvants are required here. Live attenuated virus vaccines, classically generated by passaging this in the cell culture until it loses its pathogenic properties. And it causes a very mild infection or injection. Protein-based platforms, they use the protein purified from the virus or virus-infected cells or recombinant proteins or virus-like particles. So these structural viral proteins, they're necessary to form a viral particle, but they lack the viral genome or the structural proteins. Here also we have the requirement of addition of adjuvants here. So some of the success stories with inactivated virus and live attenuated virus are the vaccine development against hepatitis A, flu, polio, rabies, measles, mumps, rubella, rotavirus, chickenpox, yellow fever. So all of these are the success stories with inactivated and live attenuated vaccines. But it has certain limitations. That is, large quantities of the vaccines would be needed to grown under the biosafety level 3. So for the whole inactivated vaccines to be developed, extensive safety testing is required to ensure live attenuated vaccines are safe and do not easily revert to the wild type. Okay, and several recombinant proteins are needed to produce simultaneously for virus-like particle vaccines. So there are some limitations for these classical platforms to be used in the vaccine development. But in spite of this, there are several companies and several institutes who are working towards a development of the vaccine for COVID-19 using these classical platforms. They are working either single or in collaboration with each other and trying to develop these vaccines using this classical platform. Because there is advantage with the classical platform, that is their inherent immunogenicity is quite strong, and their ability to stimulate the toll-like receptors are, they are very much able to stimulate the toll-like receptors, like the toll-like receptor 3, 7, 8, and 9. So this is a reason, this is the advantage of it. So that's the reason why many of the institutes are working with these classical platforms also. So next generation vaccine platforms, most of the COVID-19 vaccines which have reached the clinical trials, they're currently using this next generation platforms. So advantage of next generation platform is they can be developed based on the sequence information alone. That is, you don't need to culture the vaccines, or you don't need to work with the viral agent as such in the lab. So based on the sequence information alone, the vaccines can be developed, and they're highly adaptable and speeds up the vaccine development. So coming to the next generation vaccine platforms, we have the viral vector vaccines. These are the recombinant vaccines, often attenuated to reduce its pathogenicity. There are two types here, again, replicating and non-replicating. So replicating viral vector vaccines, they infect the cells in which the vaccine antigen is produced, as well as more infectious viral vectors are also produced. Okay, so they infect new cells, and in this new cells, there also the vaccine antigens will be produced. Okay, so here in case of non-replicating viral vector vaccines, the viral these vaccines enter the cells and produce the vaccine antigen, but no new viral particles are formed. So these this platform is used by many of the academies and industries, and about 18 of the non-replicating viral vectors and 17 of the viral replicating viral vector vaccines are under preclinical evaluation. So seeing the in detail about the molecular functioning of the replication deficient viral vectors, which is most commonly used among the two. So here you can see the infectious the agent which is entering through the receptor on the cell surface. In the cytoplasm, the genome of the viral genome is released. This viral genome has the ability to synthesize or translate the viral structural protein 1 and the vaccine agent. But the host genome as such, it produces the viral structural protein 2. So all this, the viral vector protein, structural protein 1, the viral structural protein 2, and the vaccine antigen, they form an assembly, and the entire virus will be released. And when it enters into the vaccine cell, it produces only the vaccine antigen and the viral structural protein 1, wherein the vaccine cell do not have the ability to produce the viral structural protein 2. So these antigens are produced, which when it comes in contact with the immune system is activated to produce the antibodies or the cell-mediated immunity. So that's how this replication deficient viral vector vaccines work. So there are several companies which have used this platform for its development of the vaccines, and so advantage is here that as a result of endogenous antigen production, both humoral and cellular immune systems or immune responses are stimulated, and single dose can be sufficient for the protection of the person. Okay, so we don't need multiple doses, because suppose if there is no enough stimulation of the immune system or no enough antibodies which have been synthesized, then you require multiple doses. But in this system, we can the single dose can be sufficient for the protection. So next, when we moving about with the subunit vaccines, these are acellular vaccines, since they do not contain the whole viral structures, contain only the immunogenic part of the whole virus, quite safe and convenient, but it can show low efficacy due to presence of unfavorable epitopes, and about 50 subunit candidates are in the pipeline for the development of the SARS-CoV-2 vaccines. For the subunit vaccines, we have to identify the antigen which towards which the vaccine can be developed. So we have the antigen selection in this SARS-CoV-2, that is the spike and spike protein, nucleocapsid protein, M protein, and the E protein. Among all these, the spike protein is the one which is taken up by most of the viral developing academies and the industries. So they are working towards developing the vaccine against the spike protein. I'm not going to details of this, because I think in yesterday's talk, the entire details of the spike proteins, the M proteins and E proteins have all been discussed. And just to mention here, the diagnostic assays which we use are mainly designed against this nucleocapsid protein. Okay. So there are several companies who are working with this platform of subunit vaccines. And this is a type of vaccine which is safer and easier to produce, but often requires addition of adjuvants to elicit a strong protective immune response. So that is about the subunit vaccines. Next, coming to the nucleic acid based vaccines, we have the DNA and mRNA vaccines. So DNA vaccines, this is a synthetic DNA construct encoding the vaccine antigen. So here the production does not require the growth of the live viruses and can rapidly be upscaled to in response to emerging pandemics. Like this has been used against in the pandemic of influenza also. For efficient uptake of the construct into the cells, the injection needs to be followed by electroporation, and the vaccine antigen is expressed from the DNA construct which has been injected. Coming to the mRNA vaccines, the nuclear translocation and of the DNA construct and transcription into mRNA can be bypassed by using mRNA vaccines. Okay, while using DNA vaccines, we have to ensure that this DNA vaccines are transported up till the nucleus and then it is transcribed to the mRNA. But with mRNA vaccines, this step can be avoided. So the self-replicating RNA vaccines induce protective immunity using lower dose. And these constructs include modified nucleosides to prevent degradation, because when compared to DNA, mRNA vaccines are not that stable as compared to the DNA vaccines. DNA being double-stranded, it is quite stable. But mRNA, since it is single-stranded, it is not very stable when compared to the DNA. So here we have to have the constructs need to have modified nucleotides so in order to prevent degradation. And carrier molecule may be necessary to enable the entry of the mRNA into the cells. So DNA or mRNA vaccines can be adapted quickly when new viruses emerge, and they induce both humoral and cellular immune responses. But the disadvantage is that multiple doses are required here. So this mechanism of action of the DNA vaccines, you can see here, once the DNA vaccines are injected into the cells, they are taken up by the host genome, and there will be the synthesis of mRNA using all the host genome tools. And this mRNA it is processed by the Golgi apparatus and the endoplasmic reticulum, and it is presented to the T cells on the MSC1 complex. The T cells, once it comes in contact with the antigen, then the immune system is activated and cellular immune responses are seen. It can also be presented to the helper T cells on the C2 complex, and these helper T cells respond in the way of releasing the cytokines. Okay, so the cytokines can come and stimulate the B cells. This B cells, upon stimulation, will release the antibody which is specific to the antigen. Also, there will be direct stimulation of the B cells by the mRNA which has been synthesized from the DNA construct. Okay, so this is how the DNA vaccines work when it is injected into the cells. Coming to the mRNA vaccines, we have again non-replicating mRNA vaccines and self-amplifying mRNA vaccines. So in the non-replicating mRNA vaccines, it encodes the coding sequence, okay, and it has a 5' untranslated region. It has got a 3' untranslated region, the 5' capping, and the 3' poly A tail. When comparing the structure with the self-amplifying mRNA vaccine, along with all these sequences, it also has an additional sequence for replicase. Okay, so upon injection, both of them have the similar route of entry. It is taken up by endocytosis into the cells, and when coming to the self-amplifying mRNA, it is presented to the ribosomes, and then the replicase which is present in the construct of the mRNA, that helps in amplifying the mRNAs, and then it induces the synthesis of more number of antigens. Okay, when compared to the non-replicating mRNA, so there is no amplification of the mRNA as such, and there will be the antigens which are produced are then expressed on the cells and triggers the immune system. Okay. So coming to the COVID-19 vaccines, companies working with this kind of platform, many companies when compared to the other platforms are working on this DNA and mRNA platform vaccines. And These RNA vaccines, they are quite safe, they are non-infectious, efficient in eliciting the reliable immune response with faster and cheaper production, quickly compensate for the urgent scalable demand against the COVID-19 pandemic. So along with all these platforms, we have something, the nanotechnology based approaches, which because all the platforms are there, but delivering this vaccines into the body, the delivery system also has to be very efficient, so that the vaccine goes and acts at that which are required for triggering the immune system. Okay, so we have here the nanotechnology based approaches, which offer solution for to delivery the vaccine by trafficking the vaccine to appropriate cellular population and the subcellular locations. So the many of these nano platform technologies which are used, like cationic nano emulsions, liposomes, dendrimers, and polysaccharide particles have been employed. So though I'm not an expert in the nanotechnology, but this is also been used, tried, so that these vaccine candidates which have been designed for the coronavirus can be efficiently been delivered to the right population of cells and at the right subcellular location. So this kind of these kinds of tools are very important for us to have a successful vaccine. So the latest data of how many of these academias and industries are working towards the development of vaccines. Some of the academies are, the 13% of the academies are working alone, and the industries which are working in isolation, and some of them working in partnership. Okay, and the various platforms each of them are using. So we can see here the subunit vaccines and the viral vector vaccines are the one which are commonly used by many of these vaccine developers. And some of the vaccines which have entered into the phase 3 and phase 2 trials, here also we can see that the RNA platform has shown some success rates, and even the non-replicating viral vector vaccines have shown some success rates in coming to the phase 3 trials. And there are n number of vaccines which are there in the pipeline for in the exploratory phase and the preclinical phase and the phase 1 stage. So all these institutes or academies or industries which are working towards this development of the platform, they are all at their tip of the toes to come up with the successful vaccine. And the latest list of the vaccines which have entered into the phase 3 trials. So among them, we have here the University of Oxford, the AstraZeneca vaccine, which is the technology transfer has happened to the Serum Institute of India, which has entered the phase 3 trial. Of course, maybe 2 days back, we have heard that this trial has been paused. Probably they're investigating for whatever adverse event has been happened to one of the candidates who has taken this vaccine. This trial, the phase 2 trial, has our hospital, JSS Hospital, also has been registered for this trial, and we are also one of the candidates who have received the vaccine. So there was a screening process which happened, and we were screened for the COVID negative testing, that RT-PCR negative testing, as well as our serum antibodies, which had to be negative. So once the screening process was completed, then we received the first dose of the vaccine. It is a randomized controlled trial, so it could be a vaccine or the placebo which we have received, which we are not sure of what we have got, because the person who's giving us the vaccine, as well as we who are taking the vaccine, also were unaware of what we have got. But of course, it's been about 14 days since I have been injected with the vaccine, and no adverse effects has been seen so far with me. And another vaccine which we are also part of is a BCG vaccine, which will be shortly started in our hospital. And along with that, there are many other, this is just the first page of the tabular column, which I have placed here, because there are about 10 pages like this, which have been updated on the WHO website, and all of them are in different stages of clinical trial. So this is a global distribution of the clinical trials by phase. So you can see here most of the trials which are happening in the United States, the European countries, in China, and the 2 trials in India. 3 trials are happening. One is the Covishield, which is from the Oxford Medical College, and the indigenous vaccines which have been produced by our country, that is the Covaxin, and there's one more which is produced by the Zydex Cipla company. So these are 3 vaccine trials which are happening in India. So what are the challenges when we are in the phase of this development of vaccines? So there are several challenges which we have to face. It is inadequate preclinical data and lack of detailed information on the protective correlates of the immunity. Some vaccine formulations may trigger undesirable cell-mediated response or antibody-dependent disease enhancement. So this also we have to be watchful for. Vaccines are to be used in populations with less responsive immune systems. That is, once the vaccine has come up, then we have to give it to everybody in the population, either they are immunosufficient or immunocompromised. So even for the immunocompromised or immunosuppressive patients, we should be able to come up with vaccines, maybe a different dosage or something, but even they should be receiving the vaccines. There's a huge investment, apart from time and effort, there's a huge investment which has to happen for a successful vaccine development. High cost of vaccine development usually result in a premature abandonment of potentially useful products. So probably some of the institutions cannot go ahead with the regulatory licensure procedures. So they will pause the study. And there are several regulatory issues and inadequate access to vaccines in poorer countries. So this also has to be overcome. Scaling up of the manufacturing process for the winning candidates is very much important. So that also is a challenge to be faced once, even after we get the right kind of a candidate. So what is a way ahead? To find the appropriate animals or animal models that would be that would affect or it would be affected by the virus in the same way as the humans are affected. So the right kind of animal models. So study in detail the pathogenicity of this virus will be required. More human samples have to be collected and analyzed for how the antibody production happens, because in the vaccine what we have received, it is like after 14 days it's the cellular immunity which will be triggered, and it takes about 28 days for the antibodies production to start. So that way we have to analyze several human samples to see whether this kind of antibody production does happen, or how it how long the antibody lasts, and how much protection it gives when the person is again infected by the viral candidate. And we have to gain a greater understanding of the mechanism of action of the currently used adjuvants. So what are the adjuvants which are currently used, how they are, what is their mechanism of action, how they trigger the immune system. So this again has to be studied in depth. And development of vaccine delivery systems, especially for the use in immunocompromised population. So we have to also develop some of the delivery systems so that the immunocompromised population is also benefited by this. And seek conserved antigens and monitor the genetic variations of the infectious organisms in the community. So it is not that once you develop a vaccine, it is the end, because suppose if the viral antigen gets mutated, a different strain is formed, then again the whole process has to be repeated, because you have to tackle the new strain now. So this whatever vaccine you have right now may not work for the new strain. So that again is a challenge, and continuous studies, continuous research should happen so that you can monitor the infectious organisms in the community, and you're much ready for the change of the strains much ahead, so that you can tackle this situation also. And more investment is of course required for vaccine research, and that's what all the countries are doing right now. So they have invested everything. So all the fundings for all the other projects have been stopped, and only the funding for COVID is going on right now. and also facilitate the development of vaccines in developing countries. So because some of these poorer countries may not have the access to the vaccine, so we have to think about ways in which all the population, so the whatever vaccine has been developed or will be developed, it should be affordable to all kind of the population, and the entire it's not like it should be available only in the developed countries, but also the developing countries, a poorer sector of the society also should be given with the vaccines. So definitely we will get there, but it will take just the right amount of time to reach that place. So hoping that we can successfully complete the trials of whatever has been entrusted in the JSS hospital, and definitely benefit the society by and large by controlling this pandemic. Till then, I think we should follow all the process of what the social distancing and the wearing of the mask and the sanitizing until the proper vaccine is released. Okay, with this, I thank you, and over to the organizers.
Thank you, ma'am, for sharing your insight with us. Now it's time for question and answering. I request Ms. Lydia Vandana, Head of the Department, PG Department of Biochemistry, St. Philomena's College, to take over.
Good evening, ma'am.
Yeah, good evening.
Ma'am, there's a question from Dr. Ashwini. And the question is, what's your opinion on repurposing BCG vaccine for COVID-19? I repeat. Hello.
But this is not a specific to boost the immune
Madam, your voice is not clear.
So that
Can you repeat, madam? Your voice is not clear.
yeah, it is not a specific target as such. Yeah, hello.
Hello, ma'am. Yeah.
Yeah, can you hear me now?
Yes, ma'am.
yes,
Yes.
Okay, so yeah, we are also part of the BCG vaccine trial, which will be starting very soon, probably by next week in our hospital, and the recruitment of candidates for this receiving of this vaccine also will be starting. But this vaccine is kind of something to boost the immune system. It is unlike the other vaccines for like Covishield and Covaxin, which has been the trial which has been running. The BCG vaccine is not the specific for COVID-19. So it just boosts the immune system. It will help the production of antibodies and cellular mediated immunity.
Thank you, ma'am. There's another question. What is the effectiveness of plasma therapy in COVID patients? Can the same therapy be tried in treating any other infectious disease?
Yeah, plasma therapy is something like a it is not the active immune, we don't stimulate the immune system actively. It is something like passive immunity. So here, definitely there are antibodies in the patient who has encountered the infection. And after 28 days, as I told you, it takes about 28 days time for the antibody production to happen. So there will be antibodies which can help the patient who is probably in a critical condition right now. But again, here, the efficacy of this has not been completely
Yeah, from my madam side, there is some technical issues, network issues. Participant, please wait for a while. Hello, madam. I think your network is not good from your side.
Talk from here.
Yeah. Hello. Can you hear me
Yes.
now?
Now,
Yes,
yeah.
ma'am.
Okay,
Yeah.
good, madam.
Okay. Yeah. Hello.
Hello,
Yes,
ma'am.
madam.
Yeah.
So, was I audible when I answered the
No,
previous
ma'am.
question?
No, ma'am. Could you please
Okay.
repeat it, ma'am? Yeah.
Yeah. So, this plasma convalescent therapy, it is like a passive immunity which is given to the patient. As it once a patient is infected with COVID-19, it takes about 28 days for the antibodies to be developed in the patient. So then he can donate his plasma to a person who is infected and is critical. Okay, so this kind of therapy does work in few of them, and it has not shown some success rate in few others. So again, the exact efficacy of this treatment has not been ensured so far.
Thank you, ma'am. There's another question.
Yeah.
WHO has discontinued hydroxychloroquine for treatment of COVID, but still some countries are using it. So, can you comment on that?
Hydroxychloroquine, probably the speaker in the yesterday's talk would have commented better because he was talking about drug repurposing. But yeah, initially we were advised to take the prophylactic dose of hydroxychloroquine, but then due to some effects on the cardiovascular system, this drug was discontinued, and definitely it has shown efficacy in treatment of other infectious diseases like malaria, but again, in the treatment of COVID-19, it is questionable still.
Thank you, ma'am. There's a last question.
Yeah.
Does the route of drug administration play a significant role? Hello?
The route of drug admission,
Yeah, administration. Does it
is it
play a significant role?
administration? Yeah, it is doesn't actually play, because as long as it stimulates the immune system, because most of the vaccines are given by either by IM, intramuscular route, intradermal, or subcutaneous routes. But recently there is a vaccine which have been the delivery route was the intranasal spray. So there will, because this disease is something which is affecting the respiratory system,
Yes.
yeah, of course, maybe the intranasal spray will work there, but depends upon disease to disease, which is a target organ for that particular disease. Here, the respiratory system is a target organ. So probably these intranasal sprays will work there.
Thank you, ma'am. Thank you so much for kind enough to answer all our questions. Thank you so much, ma'am. Over to
Thank
Doran.
you. Thank you.
Thank you, Ms. Vandana. I now request Ms. Shruti to give the overview of the session.
Thank you, ma'am. To begin with, I would like to thank Akila ma'am for joining us through an interesting session on vaccines of COVID. Indeed, that was an interesting talk. To give an overview, ma'am gave a brief introduction on body's defense mechanism, significance of global medical emergencies caused by viral infections, and need of awareness, followed by decoding of genomic sequence of novel coronavirus. She highlighted the multiple potential targets for SARS-CoV-2 vaccine development, which involves computational platform and molecular docking studies, advantages, and various challenges in vaccine development. We all appreciate yourself being a part of phase 3 trial of COVID vaccine. I once again thank you, ma'am, for being a part of our webinar amidst your busy schedule. Thank you.
Thank you.
Wait.
Thank you, ma'am. I once again thank our speaker, Dr. Akila Prashant, for being with us today. Thank you once again. We now break for 15 minutes and resume back at 5:30 for our next session. Please stay connected with us for the next session. Thank you once again.
Hello, sir.
Yes.
Yeah. Good evening, sir. Can we go to test your screens? Yes, presentation.
Yes. So
Yeah.
should I go for share screen?
Yeah, yes, sir. For testing purpose, sir.
Yeah.
Yes, sir. It is visible now.
Visible.
Yeah, it is visible. Yeah.
You can see change, right?
Yeah.
Okay.
Yeah. Just
So
stop,
should I-
sir. Yeah, stop it, sir.
Okay.
The college started postgraduate courses in the year 2008 in chemistry and social work. After becoming autonomous, it added few more courses in all disciplines. Today, the college has 11 postgraduate programs under choice-based credit system pattern. The courses are designed to give weightage in core papers to learn more of skills, supported by soft core papers and open elective from other departments to enhance students' knowledge beyond their program. The University of Mysore has recognized the chemistry, social work, journalism, and physics departments as research centers. 4 students have enrolled for their PhD program. To provide better facilities, an independent postgraduate center was established with well-designed labs, library, classrooms, and multimedia projectors with internet connectivity and uninterrupted power backup. The overall program outcome of postgraduate courses is to train the students to acquire knowledge leading to innovation, entrepreneurship, new technologies backed by effective communication skills. The curriculum of the programs is regularly revised as per the need of the society. Some of the major innovations introduced in the curriculum are open electives, interdisciplinary papers, and Swayam MOOC courses. The students are trained through blended teaching with flipped classes. The students and faculty members continue to enrich their knowledge by organizing and attending seminars, workshops in the college. To inculcate research culture among the staff and students, the department have introduced project works leading to publication of original research articles in reputed journals. The college is also bringing out a biannual St. Philo's multidisciplinary peer-reviewed research journal with ISSN number to publish their research articles. The faculty members have completed UGC-funded research projects. The management also supports with seed money to carry out research.
Welcome back. We appreciate you being here with us. We now have our second session by Dr. Akula Narasaraju, Assistant Professor, Department of Physiological Sciences, Oklahoma State University, Stillwater, USA. He's going to present on the topic viral replication and its interference with host immunity. I now request Dr. Shiv Kumar, Associate Professor, PG Department of Chemistry, St. Philomena's College, to introduce the speaker to us.
Thank you, madam. It is my pleasure to introduce Dr. Akula Narasaraju. Sir has PhD from Osmania University in the year 2001. His education also includes postdoctoral studies from Oklahoma State University, USA, and research fellow from National University of Singapore. His scientific accolades include development of preclinical models of evaluation of combination therapy for influenza and COVID-19. His research interest includes influenza virus pathogenesis and therapy, SARS-CoV-2 pathogenesis and therapy, bacterial pneumonia. Long-term goal of his research are to investigate and develop novel drug treatments that can be utilized for the treatment of acute respiratory distress syndrome, pathophysiology in different viral and bacterial infections. To his credit, his virology lab was the first to identify that excessive neutrophils recruited during influenza aggravate pulmonary damage through induction of neutrophil extracellular traps and the release of extracellular histones. Currently, Sir and his team are testing combination drug treatment and that targets host pathology and virus growth in murine and pig influenza models. He has good number of research publications related to SARS-CoV in the journals of prominent repute, to name a few: Frontiers Pharmacology, Annals of Internal Medicine, American Journal of Pathology, American Journal of Respiratory Cell and Molecular Biology, Current Molecular Medicine, and the list goes on. Sir has 40 publications in the journals of international repute. His H index is 22, I-10 index is 27. It is a warm welcome you, sir. Over to you, sir.
Hello, can you hear me?
Yes, sir. You are.
Yeah.
You-
Thank
you are
you
audible.
for your kind introduction. So now I'm going to do the share screen. So is it visible now?
Yes, sir. Visible, sir. You can go ahead.
Yeah. Okay. Thank you. So for past 2 days, I think you have been listening to many lectures on COVID, host immune response, as well as various drug treatment strategies, and also vaccine development strategies. So I'll be more focusing on what happens when the virus infects the lung. So the interaction between the virus and the host cells in terms of immune response. And my focus will be mainly on influenza viruses because I've been working on these viruses for the past 20 years. I just started working on coronaviruses. But indeed, while I was working in Singapore, I also worked on SARS coronavirus 1, which occurred in 2003 for about 1 year. So if you take the viruses in general, they are classified based on the genome structure. So basically divided into two types: DNA viruses and RNA viruses. And they're further subdivided based on the presence of envelope or presence or absence of capsid uh, of these viruses. So since I'm going to talk on influenza viruses, so these viruses basically belong to a negative sense RNA group and belongs to the group called Orthomyxoviridae, so which contains the segmented RNA genome. And if you see next to that is a positive sense RNA containing viruses group, That's where the coronaviruses belong to. So I'll be talking mostly on influenza viruses, maybe briefly touch on COVID or coronaviruses also. So influenza viruses also now we have been facing very hard time for the past 6 months. The whole world is completely. I mean, if you see anyone talking, they're mostly talking about the COVID-19 because this particular virus is causing so much devastation, killing thousands of people every day, and causing so much economic loss, as well as it's also so contagious that it is infecting a mass. No, it's a mass. It's causing mass infections. And if you look at the history, this is not the first time where we have experienced pandemic situation. If you go back almost 100 years, more than 100 years ago, this is when the first pandemic occurred by influenza viruses. It's a type of avian influenza virus that outbreak occurred immediately after the World War One. So after the World War One, the American army returning from different parts of the world, especially from Europe and Asia, they started falling sick with flu-like symptoms and succumbed to infection. So soon they realized that there's a flu outbreak. So within few months, there are thousands of deaths reported in America and some parts of Europe. And this virus spread every nook and corner in a span of 2 years, killing about 50 to 100 million lives. So imagine, so far, if you see the COVID-19 killed about 1 million, it's not even 1 million. Imagine in 100 years ago, this virus, this particular influenza virus killed 50 to 100 million people. So it was like 1/5 of the world population perished in a span of 2 years. So this tells you like how devastating these viral pandemics could be, especially the influenza or now we are experiencing SARS coronavirus. So it did not stop there. So after 1918, every once in a decade, these viruses are causing a pandemic-like situation. So for example, Asian flu occurred in 1957, killing about 2 million people. Similarly, Hong Kong flu that occurred in 1967, as well as the Russian flu in 1977. So each time when it originated as a pandemic, it's killing about 1 to 2 million people. So that's what we see here, the situation in case of COVID-19 also. And if you see the newspaper during that time, so this is exactly what we see today. Funerals are banned, schools are closed. So everything stops. No jobs, no academy, nothing. I mean, so this is the situation I think you face one time in a life, I would say. So besides this Spanish flu or even Spanish flu outbreaks, so there was first outbreak of bird flu or avian influenza occurred in 1997 in Hong Kong. It was in a small poultry farm and killed about 18 people, especially the poultry farm workers. So thereafter, these avian influenza viruses also causing epidemics. About 40 to 50 epidemics also reported within the Southeast Asia, and there were only about 300 to 400 deaths. But compared to millions of deaths, the death number is extremely low. But the scientists are so much concerned about these avian influenza viruses because some of these strains, especially the Vietnam isolates as well as Thailand isolates, they can kill human within a week. So they don't even allow the adaptive immunity to kick in. So I happened to attend a lecture by Dr. Robert Webster, who's considered as a father of influenza research. He's been working on influenza viruses for more than 65 years. So during his presentation, he just mentioned that some of the strains of bird flu are so nasty that he has never seen such a virulent pathogens. So the excuse me. So the only we are so lucky that these viruses lack human to human transmission ability. So if you see in case of COVID, so COVID is so contagious. So if there is one infected person attends a meeting, all the people that are present around, they get. So whereas even the person infected with bird flu attends a meeting, there will be no cross contamination. So the reason is this particular virus lack human transmission ability. So that depends on the receptor structures that we have in our lung. But similarly, swine influenza virus pandemic that occurred in 2009. So it was originated in somewhere in Mexico in a pig farm, and soon it spread every nook and corner of the world within 2 months, killing about 220,000 people. It is still circulating in some parts of Asia. So like that, if you see, there are so many pandemic outbreaks occurred due to influenza viruses. And coronavirus, when it comes to coronavirus, there was outbreak occurred in 2003, especially that the outbreak was very much geographically focused on on Singapore, Hong Kong, and China. It did not spread to other parts of the world. But whereas the current uh, COVID-19 or SARS coronavirus 2, also it was originated in China. It spread within 2 to 3 months to every part of the world, and it is causing so much devastating infections. So today we'll discuss like what happens when the virus enters into the host, especially in the lungs. So the interaction that takes place between the virus and the host cells. So before that, uh, let me explain about the respiratory system that we have. So our respiratory tract can be divided into two parts, upper and lower respiratory tract. So the upper respiratory tract mainly consists of nasal, pharynx, and larynx, whereas the lower respiratory tract starts from trachea, bronchioles, and small bronchioles. Finally, the terminal bronchiole that opens into alveoli. If you see on the right side corner slide, so this is the typical structure of the alveoli. So these are the breathing areas in the lung. So the alveolus, if you see this alveolar structure, there are majorly two types of epithelial cells. So type 1 and type 2 epithelial cells. The type 1 epithelial cells, which are cuboidal and present in the corners of the alveolus, whereas type 1 epithelial cells, they cover 95% surface area of the uh, alveoli. So this is where actually when you breathe air, so this is where the oxygen exchange takes place. And if you see that all the red spots, they represent the capillaries, the pulmonary vasculature. So this is where actually the oxygen intake and the carbon dioxide release takes place. And the type 2 pneumocytes produce surfactant lipids, which are important to prevent the collapse of the lung during exhalation. Whereas type 1 pneumocytes have very thin cell membrane, and below the type 1 cell, there lies the capillaries of pulmonary vasculature. That's where the oxygen exchange takes place. And both influenza and coronavirus 2 primarily target type 2 pneumocytes. So you can see the structure of alveolus is so thin, so it can be highly fragile when there is even the minor damage. And these viruses cause massive death in alveolar type 2 pneumocytes as well as type 1 pneumocytes. So this results in complete collapse of the alveoli. That's why if you see the severely infected COVID patients, they get hospitalized and require the ventilation therapy. So that's because the collapse of the alveoli takes place. So this impairs the gas exchange in the lungs. So patient eventually dies with the pathologic symptoms of acute respiratory distress syndrome. So my entire research for the past 20 years focused on these cell types and see how various drug development strategies to prevent the uh, alveolar damage, as well as uh, we are trying to find various factors that promote the repair of the alveoli. So the normal epithelial regeneration, which is critical for the patient to recover from severe pulmonary pneumonia. So that's the main focus of my research. So having said, I'll go back to influenza viruses. So as I said, influenza viruses belong to Orthomyxoviridae family, which contain a segmented negative sense RNA as their genome. So the genome of influenza consists of 8 RNA segments, which encode at least 12 proteins. So based on the antigenic nature of the internal proteins of the virion, that includes matrix and nucleoprotein, these viruses are classified into 4 types: type A, B, C, and D. Further, type A viruses, which mainly cause human infections, for the subtype based on the antigenic nature of the surface proteins of the virion, that includes hemagglutinin and neuraminidase. And there are about 18, 18 different HA and 16 different NA types of identified. And these influenza viruses, they enter the host cell or the respiratory epithelium through specific receptors called sialic acid receptors. There are 2 types of sialic acid receptors: alpha 2,3 linked sialic acid receptors, as well as alpha 2,6 linked sialic acid receptors. So as I said, the avian influenza or bird flu lack human transmission ability because they have high affinity towards alpha 2,3 linked sialic acid receptors. And if you look at our respiratory tract, we lack alpha 2,3 linked receptors in the upper respiratory tract. So even if you breathe the virus particles into the lung, so even if they deposit in the nostrils, they will eventually cleared by macrophages. So they won't cause infections in the upper respiratory tract because they lack our nasal epithelium lacks alpha 2,3 sialic acid receptors. Whereas in case of COVID-19, if you see the receptor, which is ACE2 receptor present in the entire respiratory tract, perhaps the reason why COVID-19 is causing so much uh, virulent infections and it is spreading very effectively. So once the virus finds its receptor, it makes entry into the host cell and starts replicating. So if you look at the replication cycle of the virus, there are mainly 4 stages. First, entry of the virus. But in case of influenza here, entry occurs through the endocytosis. So once the virus binds to the sialic acid receptors, it undergoes endocytosis. So now the virus is present inside the cell within the endosome. Then the next step is the virus fuses, a viral membrane fuses with the endosomal membrane, uh, thereby releasing the viral RNA complex in the cytosol, which eventually enters into the host nucleus. Now, once it enters the nucleus, it starts uh, transcribing the various viral proteins. So after the formed mRNAs, they enter the cytoplasm for translation and synthesize the new viral proteins. And during that time, again, the virus undergo replication and form, because since the influenza virus contains 8 RNA segments, so each segment replicates separately. So after all replication of all viral RNA segments, they pack again with the help of some of the proteins that are translated in the cytosol. They again enter into the nucleus and they start forming packaging and again entering into the cytoplasm, reaching to the cell membrane. So that's when the hemagglutinin and spike protein expression get ready for the synthesis of progeny virus. So the assembly of new virus particles at the plasma membrane and the virus bursts out from the infected cell, and then it spreads to it infects the neighboring cells, and it keeps going. So this is what happens when the virus enters, the virus finds its target cell. So when the virus infects the host cell, there's a lot of conflict that occurs between the virus and the host cell survival and growth. So if you see the virus point of view, virus tries to overtake the host machinery for its progeny virus synthesis. So to do that, actually virus uh, tries to keep the infected cell alive for some time until it completely all the progeny virus synthesized. So that's very important because if the host cell dies, eventually the virus dies inside the cell. So it will try to prevent or delays the apoptosis in the infected cell so that the virus, enough number of virus progeny viruses particles are replicated. So once everything is done, then anyhow the cell dies. So in the meantime, virus tries to keep the cell alive and try to overtake all the machinery for its own virus replication. So from the host cell point of view, the host cell try to prevent the virus replication. So the best is to undergo suicide. The suicidal death is also called apoptosis. So that actually eliminates the growth and spread of the virus. And besides the host cell, there are several molecules, several receptors on the host cell that sense the presence of virus. They immediately activate various signaling cascades. So that prevents the virus replication at either RNA synthesis level or protein level or even at the release of the virus. And host cell also tries to produce various cytokines and chemokines that attract more number of innate immune cells, such as macrophages and neutrophils, which are critical for clearance of the virus in the infected lungs. And of course, the host also develops the adaptive immune response, where it starts producing B cells, which synthesizes antibodies against a specific virus, as well as various cytotoxic T cells that can identify and kill the virus infected cells. So all these events, in all these mechanisms, the host cells they try to limit the virus growth as well as prevent the virus spreading. So that in case there is a lot of conflict that occurs, especially the first week of infection. So that's why if you see in COVID-19 situation, so 90% or more than 90%, 95% patients are recovering. So that means this conflict, during this conflict, the host is winning. So either clearing the virus or by undergoing apoptosis or killing the virus directly using the innate and adaptive immune arms. So the patient falls sick for a week, and by second week he starts slowly recovers, and by third week he completely gets back normal. So during this 1 to 2 weeks, this conflict between the virus and the host cell takes place. So virus tries to overtake the missionary, but the host trying to prevent the virus replication as well as the spread of the virus. So as you can see, I think this chapter is covered yesterday. So I'll just briefly go through. So the primary host defense is the in, uh, either innate or adaptive immunity. Again, when it comes to innate, either it could be it could be by cytokine production, for example, interferons, or cellular responses, macrophage, natural killer cells, as well as neutrophils. And when it comes to adaptive immunity, T and B cells. But if you see at the bottom, so the innate immune response occurs immediately after when the virus enters our body. So our respiratory system immediately recognizes the presence of the virus, and they start inducing the innate immune response involving various cell types, cytokines, and also complement system. So all these they try to limit the virus replication at very early stage. So sometimes what happens if you inhale the significant doses of virus. So the innate immunity is not enough to completely remove the virus at the early stage. During that stage, the virus takes the opportunity and enters the different cell types of the lung and start causing damage. So by second or third week, the whatever the macrophages and dendritic cells mainly involved also connect between the innate and adaptive immune response. So they take the viral antigens and they present to synthesize both B and T cell types, which helps during the adaptive immunity phase from second week to it goes on. So some of the cells they go into memory and they give long-term protection. So when it comes to the innate immunity, the major host factors are that can limit the virus replication, the cytokine response, as well as the inflammatory cells such as macrophages and interferons. So when it comes to cytokines, interferons play a major role. These interferons produced by macrophages, epithelial cells, or cytokine T cells have antiviral activity. And these are small protein molecules based on the receptor specificity. They are divided into three types: type 1, type 2, and type 3. And one of the important interferons, type 2 interferon, which is interferon gamma. So if you look at how interferon signaling actually limits the virus replication. So when the virus infects a cell, the cell start producing the interferon genes. So once the interferon gene transcribes and translates and produces interferon, these interferons releases from the infected cell, and they sense the interferon receptors in the neighboring cells, and then they start activating, inducing the antiviral activity. Basically, they act by two modes. One, by induction of oligo A synthetase, and second, by induction of protein kinase. So if you look at, so when the interferon binds to interferon receptor, they activate the synthesis of 2,5-oligo adenylate synthase, so which in turn form 2,5-oligoadenylate. So 2,5-oligoadenylate actually enhances the endonuclease activity in the cell. So endonuclease activity is, by name itself, you can tell that it cleaves the RNA or DNA. So in this case, here they induce the endonucleases that are specifically for degrading RNA. So that degrades the replicating viral genome. Thereby, it inhibits the virus replication at the RNA synthesis level. On the other hand, the interferon also enhances the protein kinase expression. This protein kinase phosphorylates eukaryotic initiation factor 2, which is required for translation of the viral protein. So once this EIF2 or eukaryotic initiation factor is inhibited by phosphorylation, that inhibits the synthesis of viral proteins. So here the interferon actually inhibiting the virus replication by two modes: one, either by degrading the viral RNA or by inhibiting the translation of viral proteins in the host cell. So besides this cytokine, the host cells also undergo various mechanisms, undergo various modifications to prevent the virus growth. So one of that is apoptosis. Apoptosis is nothing but it's a suicidal mechanism. It's also called programmed cell death. So the main proteins involved in apoptosis are the caspases, which contain the cysteine, different based on the cysteine structures that are organized in the caspase, in the protein structure of the caspases. They are like different types. Basically, there are two types: initiator caspases and executioner caspases. Initiator caspases, they require to activate to initiate the apoptosis process, and they also activate the executioner caspases. And these executioner caspases actually degrade the various proteins in the host cell. So what happens when the influenza virus infects the host cell? One of the very well characterized mechanism is fast mediated apoptosis. So the infected cell expresses both fast as well as fast ligand. So when this fast ligand binds to fast receptor, the cell signals to undergo apoptosis. As you can see on the right hand corner, we found the apoptotic cell death and the lung epithelial. I think if you remember just few slides before, I've shown how the alveolar structure looks like and the presence of alveolar type 1 reticulocytes. As you can see here, the brown spots indicates the cells undergoing apoptosis. So we perform the tunnel assay where we label so during apoptosis there will be a degradation or cutting of DNA into specific fragments. The free cut ends are labeled with a specific dye. So that's what you see here. So when the cell undergoing apoptosis will be immunostained using a specific dye using the method called tunnel assay. As you can see here, there are various epithelial cells, both type 1 and type 2 epithelial cells undergoing apoptosis when we infected with influenza virus. So these tissues are from urine model. So if you look at how the caspase activity takes place, so as I said, there are two types of caspases. Initiator caspase as well as excretional caspases. The initiator caspases, generally caspases are present in the proform because if they are active, they start degrading the host cell because they're not specific to the host or pathogen. So that's why most of them they are present in their proform. So they require activation to become active and start degrading the proteins. So the whole process of our apoptosis begins with activation of initiator caspases. So once there is a signal from the like a fast signal in the cell, the procaspases starting becoming active, and they form a heterodimer structure. And once they form the heterodimer structure, they become active. And these initiator caspases, they further activate the excretional caspases. These excretional caspases kills various proteins of the cell, and cell eventually degrades all the subcellular organelles, thereby ultimately it undergo apoptosis. So for example here, so when fast binds to fast ligand, or we call it fast death receptor, it forms a fast, fast associated death receptor domain. So this fat formation is the primary and critical step in the apoptosis process. And this fat recruits the procaspases such as procaspase 8 or 10, forming a disc domain. So once the disc is formed, then this activates the executioner caspases, and these executioner caspases in turn degrade the various proteins both in the cytoplasm as well as the nucleus, and ultimately forming apoptotic bodies. So when the cell releases apoptotic bodies, and these apoptotic bodies still contains the degraded product inside the plasma membrane. So it's kind of a vesicle, and these vesicles, they will be captured by macrophages and neutrophils, and they get cleared. So it's a continuation from apoptosis. The next phase is the phagocytosis. As the cell undergo apoptosis and form apoptotic bodies, the macrophages come into picture, and they recognize. So during the apoptosis, what happens? The cell has phosphatidylserine predominantly present in the inner leaflet of the plasma membrane. So during apoptosis, the membrane flips back and exposes the phosphatidylserine to the extracellular space. So this exposure to The serine actually acts like a signal eat meal. So this gives the macrophages and neutrophils they to recognize these apoptotic bodies and phagocytose and clear from the system. So now the major question is, okay, now when you have the infection with the COVID or influenza, the cells undergo apoptosis. That is very common, as you can see in the previous picture. Then if you look at the image here, the control lung, as you can see, the intact alveolar architecture as well as the bronchiolar epithelium, everything is intact. You don't see any abnormality. You can see red capillaries also. But on the right image, you can see influenza infected lung. Complete devastation, complete destruction of alveolar architecture. And there's a lot of cellular debris. So now the question is, when there is so much apoptosis going on, why there is so much debris? Fortunately, these both influenza and COVID, they both can also kill these scavenging cells or macrophages. So thereby, there will be a natural depletion of macrophages, especially during the early phase of infection. So even if the epithelial cell undergo apoptosis, there are no macrophages to come and clear phagocytose and clear from the system. So these apoptotic cells eventually die by necrosis, releasing all the cellular toxic components into the lung alveolar space. So that gives a major trouble, especially leading to acute lung injury or acute respiratory distress syndrome in case of these both influenza as well as COVID-19. So this is one of the major area like we are looking into to prevent this accumulation of extracellular debris. So I'm just telling one factor. There may be at least 100 factors or more than 100 factors that contribute ultimately to this acute respiratory distress syndrome. So, but we are trying to focus on some of the major pathways that can, so thereby we can minimize the AIDS pathophysiology in both influenza as well as COVID-19. So another mode how the cells try to prevent the virus growth is the formation of neutrophil extracellular traps by netosis. Like apoptosis, I think you might have all learned about apoptosis as a programmed cell death, but there is another cell death mechanism also called netosis. Like similar like apoptosis, is also programmed cell death discovered in neutrophils in 2004. So it's very novel compared to the mechanism studied compared to apoptosis. So, uh, during the netosis, what happens? The cell undergoing netosis, it bursts and release the entire extra neutrophil, entire DNA chromatin into the extracellular space. So all the chromatin fibers, they release into the extracellular space, forming a net-like structure, as you can see the left-hand bottom. So these chromatin strands, which are nothing but the DNA fibers, they carry the toxic components, granule proteins of the neutrophils, that include neutrophil elastase and myeloproliferases and latex and endoprotease. These gnats, when they release into the extracellular space, they can trap and kill the pathogens in the extracellular environment. So it's a kind of a defense strategy wherein the neutrophils undergo suicidal death and kill the pathogen in the extracellular space. So here, actually, this paper we recently published in case of Francisella tularensis, which is a very severe bacterial infection. So during Francisella infection, you could see induction of gnats, and you can also see gnats trapping the bacterial pathogens. So if you look at the gnats, they mainly contain the chromatin structures, so DNA fibers. So DNA is nothing but you will have DNA as well as histone proteins, which are important for nuclear organization. So histone proteins, there are majorly 5 types: H1, H2A, H2B, H3, and H4. They form automeric structure through which, on which the DNA wraps around. So this is the typical structure you see in the when the DNA is present inside the nucleus. So these histones, they are so important for organization of the nuclear structure or DNA structure inside the nucleus. But if once they release into the extracellular space, these histones are so toxic that they can kill, they cause extensive damage to the host system. So we tried to do an experiment to take the pure histones and inoculate it intranasally into a normal mouse. So we tested like different concentrations of histones, starting from 25 microgram, 50 microgram, 100, 200 microgram. So when we gave just 200 micrograms of histones intranasally delivered into normal mouse, all the mice died within 40 minutes. So you can imagine how toxic these, because they're basically highly positively charged proteins. Once they attach to the plasma membrane, they cause pores in the plasma membrane. So that causes complete leakage, just increased intracellular calcium, and eventually the cell dies. So this is one of our major focus and also a treatment strategy that we are proposing. So the mechanism of how this netosis occurs is not yet clear, but one of the protein called peptidyl arginine deaminase (PAD4), which has been shown very critical for a specific modification, which is called synchronization of histones that occur during the NETs release. So the histones, they undergo some modification of citrullination, arginine to citrulline mediated by a peptidyl arginine deaminase, which is the first step for the NETs release. So we while working on influenza viruses in Singapore, so we frequently observe these structures that I have shown on the left side, the elongated structures. Initially, we thought these are some fibrin deposition, but actually, if you see, there is a cellular origin. So that time it was in 2008. So nets were discovered in 2004. So it was very naive, and there were no, there are like 3 or 4 papers published. So it took almost 2 years for us to identify what these could be. So finally, we characterized and found that these are the nets induction that occurs during severe pneumonia. Further, like I published almost more than 10 papers now showing like how the nets actually contribute to the host pathophysiology in severe influenza pneumonia in both mouse as well as pig models of influenza pneumonia. So histones, although we look at the histones at one direction, that they cause severe pulmonary pathology, but actually histones also inhibits the virus replication. So it also kills the virus directly, causing pores in the membrane of the virus particles. But the only problem comes, there is massive influx or massive accumulation of uh, histones or extracellular, nets occurring in the infected lungs. So that is the major problem. So they have both protective effects by preventing the virus replication, but on the other hand, they can kill all the different cell types. As you can see here, so the histones, various histones, toxicity and different cell types, both epithelial cells, alveolar epithelial cells as well as the endothelial cells. Yes, because significant cytotoxicity in these cells. And imagine there are tons of histones released in the lungs during, especially in the first week of infection. Uh, and last week there was a paper published that there is a significant amount of histones accumulated in COVID infected patients, especially with severe pulmonary damage. So that shows both influenza and COVID, they are more or less have high similarities in pathophysiology. And that's what actually I am now slowly focusing on, because we have developed some drugs for the treatment of influenza that can directly apply to COVID-19, especially in preventing the acute lung injury. So that's what my main focus currently, to see whether these drugs, because so far now there's not a single drug available for COVID treatment. So we are all using repurposed drug, whether it is malaria drug or Ebola drug or something hepatitis drug. So whatever the drug or influenza, even influenza drug. So we are trying to use various drugs that have been tested for different viruses and now testing for COVID-19. And they already started using like remdesivir or B705, which is now currently used for COVID-19 treatment. So these are all repurposed drugs. So similarly, now actually we are trying to target the host-mediated pathogens instead of directly targeting the virus. So due to the complexity of this disease, because it causes massive pulmonary damage, the lungs really looks like a liver, so they completely destroy the alveolar architecture. So just using one single drug is not enough. So that is the reason why we are trying to develop a combination treatment that targets both virus replication as well as the host pathology. So besides this, there is another uh, receptors which play important role in innate immunity is the pattern recognition receptors, which actually recognize or sense the viral molecules. For example, toll-like receptors in case of influenza. So if you look at this, there are various mechanisms by which the host cell try to prevent the virus growth. So one of that is when the virus infects, as I said, the virus undergo endosomal endosomal pathway to enter into the host cell. So virus presence within the endosome is recognized by toll-like receptor 7, which in turn activate MYD88 pathway, which is very well characterized pathway that leads to NFKB, a major transcription factor that's inside that that's synthesizes the pro-inflammatory cytokines as well as the interferon generation. So when the virus present within the endosome, it is recognized by toll receptors of the host cell that induces the synthesis of type 1 interferons. On the other hand, during the virus replication, there is a cap formation by triphosphate, which is essential for the virus during the transcription phase. So once when the virus enters the nucleus and starts transcribing, the cap is formed. So this cap formation is immediately recognized by uh, Rig molecule, which in turn activates through ARF mediated signaling, ultimately leading to type 1 interferon production. So if you see on the other hand, there is another novel pathway which is called inflammasome pathway. So we or NLRP3 pathway. It is not very clear like how these pathways exactly activated or how much this pathway actually contributes in limiting the virus growth. But it has been recognized that this is one of the critical pathway that is that enhances the growth, virus growth, uh, especially at the early phase of infection. So all these different signaling pathways ultimately lead to interferon production. These interferons, as we discussed about, they limit the virus replication. So another challenging that I want to share with you is the vaccine development. So despite the COVID-19 causing so much devastation, to so much trouble in every nation. So why there is so much delay in vaccine development? Why it is so challenging? Simple, why it is so challenging? Because there are about 7,000 deaths occurring every day. But despite there are so many noble adults here, like you say, take in America. Despite having so many noble areas and so much intellectual property and intellectual brain, but still we could not be able to develop one single vaccine, effective vaccine for COVID-19. Because there are two major challenges. One, the ability of the virus to mutate. So it frequently changes its structure. So we actually happen to sequence the COVID SARS coronavirus 2 uh, isolated at different time points from March to July. You could see about 20 different mutations occurred within the virus. That shows that these viruses are significantly mutating. So that is the major challenge in for vaccine development. And another thing, even if you have vaccine, if you inject a vaccine, you have to test efficacy. So because virus will have so many uh, epitopes in each protein. So some of the antibodies that are developed after the vaccination, they may just bind to the virus. They may not neutralize or kill, helps the virus to get removed from the system. So that is the key thing. So that is where actually scientists now uh, breaking their heads to see how effective these vaccines in in neutralizing the virus replication, neutralizing the virus process. That is the key thing. So like if you see in my in this slide, if you see if you take if this is the hemagglutinin or even the spike uh, uh, protein of the COVID-19. So there will be different epitopes which are recognized for generation of antibodies or T cell mediated immunity. So these epitopes, some of these epitopes, even if the antibodies bind, they may not interfere with virus entry or virus replication or virus release. So they may just bind to the virus. So these antibodies are not useful. So we need the antibodies which can neutralize. So when the antibody binds to virus, so the virus has to be cleared from the system. So that is the ultimate goal of any vaccine development. So this is the reason why it's taking so much time. Scientists are testing for efficacy of the virus, whether it is antibody mediated neutralization or cytotoxic T cell mediated cell, cell death of the infected cell. So this has to be tested. So that is what we call efficacy. So another point is safety. So you are injecting virus to you are injecting the inactivated virus or viral protein to a normal person. So if the individual falls sick by any cause, so that will become a major issue. So the reason why there is so much it is taking so much time to actually to bring the vaccine to the market. So, for example, like in last week, I saw the news that AstraZeneca, which has been developing the vaccine for COVID-19 for the past 6 months, halted its clinical trial, phase 3 clinical trials, because they found that one of the immunized uh, person severely fall severe sick. So now they are trying to find the reason whether the vaccine whether there is any vaccine related adverse effects, because that is not acceptable before the vaccine releases into the doctor. It should not cause any adverse effects. So for just one case, they completely halted the clinical trials. So you can imagine how sensitive this particular vaccine development program is. The reason why it is taking so long. Uh, so there is no shortcut, but using the repurposed drug is different. I mean, after the individual get infected and fall sick, you can administer various drugs to test, at least to see whether the patient recovers from the infection. But injecting into a normal individual is quite challenging. So the reason why vaccination is taking so much time. In some cases, I think like if you take the hepat, uh, HIV vaccine, it's been almost more than 20 years or 25 years. It is still in clinical trials. So imagine, at least takes 5 to 20 years for any vaccine to come into the market. At least for COVID, because of the urgency and urgency in the world, I think scientists are trying to bring the vaccine within 1 year. That's really, we should really appreciate the effort that is made to bring the vaccine into the market. And hopefully that will, you will see that maybe in a couple of months. Uh, Especially some of the vaccines are in the pipeline of phase 3 clinical trials, testing in individuals with various comorbidities, like having the COPD or other diabetes or other comorbidities, because those are the patients which are those are the patients who are highly succumbing to the succumb to the COVID infection. So that is when the clinical trial stage is now currently, and hopefully we'll see the vaccine in next couple of months. So with that, I stop here. I would like to take questions.
Thank you, sir, for such an interesting talk. It's time for question and answering, so I request Ms. Lydia Vandana to take over.
Good evening, sir.
Right.
Could you highlight on host immune system responses in lower organisms?
In lower organisms.
Yes, sir.
So lower organisms, are you referring to animals like?
Yes, sir. Yeah, animals.
Yeah. So there is a lot of conflict in that, because there are the data, if you look at that, so even the mouse can reproduce 95% of the host immune response what we see in humans. But the only problem is some of the proteins, they have homologous proteins, like IL-8. If you take, we'll call it CXCL1 in humans. So there is a homology, uh, but yes, you are right. But the only thing, the majority of medical clinicians, they are not happy when you test in animals and show that this works in humans. So there will be, that's why, I mean, even for vaccine development of COVID, it's going in two parallel steps. So what we are listening in news is clinical trial 1, 2, 3. But actually, before that, there is a preclinical study. So which is actually basis even to go for clinical trials. So they have tested in mice and ferrets, and maybe and hamsters, especially hamsters. So mice and hamsters, they tested the COVID vaccine first. So after they get the positive result, because when it comes to host immunity, it is 95%, I would say, very similar. There could be some difference, but the way hosts respond in terms of innate immunity, adaptive immunity response, it's all very, very similar mechanism. In fact, our understanding, the entire host immune response is based on the animal studies, animal data. We just apply that what we from the clinical samples what we see, whether it is antibody response or T cell response or T helper response, helper cell response. So preclinical model actually gives a basis to understand the host immunity before before testing whether vaccine or any drug into the human clinical trials. So I won't look at that very separately, but only thing, you need to understand how it works in humans. You will test in animals. Sometimes you test in small animals like rodents first, mouse or hamsters. Then you go to large animals like ferrets and swine. Like we tested first mice, then we went to swine to test the efficacy of the combination therapy. So if that passes, then when I published that, so immediately a company called GlaxoSmithKline took my paper as a basis to test clinical trials last year. So they published, and they gave the first reference of my publication to show that yes, this combination therapy tested in mice and swine, it is working. So now we want to test in clinical trials for humans, and they have been testing in human clinical trials for past 1 year now.
Thank you so much, sir, for answering. We are glad that you are amidst us. Thank you very much, sir.
Thank you. Is there any question
Uh,
from audience?
Sir, there's a question. As mutative property of virus, does the virus change the spike or all the enzymes?
Yeah, we found mutations at least in 5 different proteins of the virus, SARS coronavirus. As I told, we actually sequenced coronavirus isolated, like okay, I'm now currently taking in charge in Oklahoma for COVID testing. So every day we receive 1,000 to 2,000 samples. So we are testing by RT-PCR. So whatever the positive samples, we keep them separate. We try to sequence. So we found at least from 5 different isolates from 5 different time period between March to July. We picked up like 5 randomly positive samples and sequenced. We found at least 5 genes showing 20 mutations. It is not just spike. So every single gene can get mutated.
Thank you, sir. There's another question. Is COVID also attack on NET, netosis?
Pardon, there was a break.
Yeah. Is COVID also attack on NET? That is the netosis, what you spoke about it.
Yeah, actually, we formed a group from almost 8 countries now. We are meeting every 2 weeks and discussing like how the netosis is actually playing in pathophysiology of COVID-19. So we have about 70% of clinicians in the group and 30% of scientists. So we are meeting every 2 weeks. We're trying to understand. Yes. So COVID-19, there is a lot of nets infection in the lungs, but the only problem is having the autopsy samples. So some of the clinicians, they're now managing to collect the autopsy from the dead patients. And now actually we are analyzing, and our initial study shows us there is a massive neutrophil nets induction in the COVID infected lungs. So now we are trying to see various therapeutic mechanisms to see like actually in my case, I have a couple of drugs which we have tested in influenza. We can directly apply to the company. Even to tell you that I approached AstraZeneca 3 months ago with a drug that we have tested in influenza to test in COVID-19. But AstraZeneca has a lot of money for vaccine development. So actually, they have a couple of very important drugs that prevents the neutrophil influx as well as neutrophil nets formation. So I requested them that I would test those 2 drugs, AstraZeneca drugs, in COVID animal models, but they are not very encouraging because they're now busy with the vaccine development. Probably eventually I'll try to convince them, because but all we need to do, we have to publish as much as the data, the preclinical data. So that's when I'm once again telling the preclinical data is so important. That gives the primary evidence to move forward to the clinical trials. So even the AstraZeneca, first thing they ask, okay, show me any preclinical data. Then I will support you money as well as data. Since we did not have so far the preclinical model, so we could not answer their question. So they said no, we are busy with the vaccine. So we need more time to approach again for them to get involved in this.
Okay, sir. Thank you very much. We are done with the questions, sir. Thank you.
Thank you. We appreciate for giving the opportunity.
Thank you.
Thank you, ma'am. I now request Dr. Shiv Kumar to give the overview of the session.
Thank you, madam. It is my privilege to conclude the talk on viral replication and its interference with host immunity by a very erudite person, Dr. Akula Narsaraju. Sir began his presentation with a classification of animal virus. He highlighted the previous pandemic outbreaks. Sir spoke on mode of influenza and SARS-CoV and entry into the host system. He discussed the host defensive action and eliminate the virus through activating innate mechanism immunity response, discuss the mechanism of interferon mediated inhibition of viral infection. He also emphasized how host immune response is cytotoxic to the host system, leading to organ failure. He also stressed on why development of COVID-19 vaccine is challenging. I thank Dr. Akula Narsaraju for his interesting talk. Thank you, sir, for your time. Thank you, sir.
Thank you so much.
Thank you, sir. I once again thank the speaker, Dr. Akula Narsaraju, for being with us today and for sharing his view with us. Thank you, sir. We have come to the end of our international webinar. Before we go into the valedictory program, a few announcements to make. I request all of you to stay connected with us till the end of the program. The feedback form for today's session will be posted in the chat box. Kindly fill in the feedback form before you leave the meeting, as it is mandatory to fill in the feedback forms to generate your e-certificates. As we come to the end of our scientific sessions, I call upon Professor Othbert Pinto, Director, Postgraduate Studies and Research Center, St. Philomena's College, to deliver the valedictory note.
The pleasant morning to the resource person, Professor Narsaraju, and pleasant evening to all other participants. I thank the organizers for inviting me to deliver the valedictory address of this 3-day international webinar on immunity in today's world. The innovation in digital technology has made our life very comfortable. Today, without stepping out of our houses, we get everything to our doorstep except immunity and health. The COVID-19 pandemic perhaps made all of us to realize the importance of our health. During the lockdown period, we were forcefully confined to our homes. Hotel, restaurant, hospitals, and clinics were closed, and surprisingly, our health condition improved as we had enough time to do exercise and to prepare healthy food. The lesson we learned was that we should take time out from our busy schedule to give attention to our health. As the pandemic was spreading out, the market was flooded with a variety of immunity enhancing products. The people were not able to select the best one. To bring awareness on immunity, the Department of Biochemistry of our college has organized a 3-day international webinar. I congratulate the department of their excellent thought. The speakers from different parts of India and US have deliberated on the importance of immunity to prevent several infections, including the present pandemic. I have been a part of all sessions. The Department of Biochemistry has done a wonderfully to communicate the participants the importance of healthy diet and lifestyle to build a healthy immune system. Indeed, this webinar provided an opportunity to all of us to change ourselves to control our health and immunity. I extend my thanks to all the participants for their active participation in the webinar. Thank you. Stay home, stay safe.
Thank you, sir, for your kind words. Now I call upon Dr. Yashashwini, Assistant Professor of Biochemistry, to deliver the vote of thanks.
Thank you, ma'am. Good evening to one and all. Sunset is a wonderful opportunity for us to appreciate all great things the sun gives us, says Mehmet Morat Ildan. We have come to an end of this 3-day international webinar. I deem it to be my esteemed privilege to propose the vote of thanks. I take this opportunity to thank the Almighty for showering infinite blessings on us. I wholeheartedly thank our Lordship, Bishop of Mysore, Most Reverend Dr. K. A. William, for gracing and giving presidential address. Thank you, His Excellency. I like to thank our management, Rector, Reverend Father Dr. Bernard Prakash Parnas, for his continued support for all we do in the department. Thank you, Father. I thank our Vice Rector, Reverend Father Maria Xavier, Campus Administrator, Reverend Father John Paul, and our beloved Principal, Dr. Ruth Shantakumari, for their encouragement and support. I express my profound gratitude to our Director, Professor Otpat Pinto, who is a driving force for us to conduct this webinar successfully. Thank you, sir. I feel honored to convey my sincere thanks to our resource persons, Professor Cletus J.M. D'Souza, Dr. Vijay Kumar Reddy, Dr. Hemachandra Jha, Dr. Shashidhara Murthy Tawal, Professor Akila Prashant, and Dr. Akula Narasaraju for elegantly journeying us through this 3-day international webinar on immunity in today's world. Special thanks to Dr. Sukanya, HOD, PG Department of Mass Communication and Journalism, for her constant support to conduct this webinar. I fallen short of words to express my heartfelt thanks to Mr. Venugopal Gowda, Mr. Raviraj, Mr. Arun, Mr. Sunil D'Souza, and Mr. Sandesh for supporting us technically and helping us carry out this webinar successfully. I also thank to our non-technical staff for their cooperation throughout the webinar. The presence of our participant has made us glad. I thank them for their active participation in this webinar. I thank Ms. Shruti, Organizing Secretary, Ms. Lydia Vandana and Ms. Renita Ross, Coordinators, and Organizing Committee members, Dr. Dorin D'Souza, Dr. Shiv Kumar, Ms. Akshata, and Ms. Plessy, for making this webinar a successful one. Thank you, one and all.
Thank you, ma'am. Kindly note, the overall feedback link will be sent to your registered email ID latest by Tuesday, once we scrutinize the details provided by you. Thank you for being with us for the last 3 days. Hope this webinar has benefited you all. Hope to see you all again. Thank you, one and all.
Thank you, sir. Thank you, one and all. So now this webinar is